Okay, Bio 30, this is going to be the last lecture for Mendel. Then I'm going to take a break for a few days before I do molecular genetics. This is the last thing that you need to know for Mendel, so you can do everything involved in the Mendelian genetics section, right? The Mendel quiz, the pedigree analysis, the Mendel unit test once I'm done this lecture. So we're going to talk about gene linkage here. We talked about segregation and independent assortment and all that stuff. But we know that chromosomes, when they separate, they segregate and swap alleles right during crossing over in meiosis. So we talked about independent assortment and segregation, meaning that traits and alleles are not linked. We've been able to observe that in some cases, though, chromosomes are actually linked in equal predictable probability. So it doesn't violate Mendel's laws, but now that we understand the structure of DNA, we know why this happens. So this is an extension on Mendel's laws, and we're going to make chromosome maps and look at recombinant DNA in this section. So it turns out that when you look at chromosomes, okay, and when they cross over during meiosis, the deal is that right. So your centromeres are here. We know that alleles that are so from genetic analysis, so if they're further away from the centromere, they are more likely to be swapped. So here's what actually happens with crossing over in meiosis. The alleles that are further away from the centromere, they're more likely to be exchanged across non-sister chromatids. And this should make sense, right? It's a lot of energy expenditure to, break a chromosome up right at the centromere and swap this massive amount of DNA. Instead, these alleles at the ends of the chromosome are more likely to be swapped. So there are not equal frequencies by which alleles swap. So independent assortment and segregation do work, but there's a caveat to separation based on your distance from the centromere, right? So there are higher probability alleles that do get swapped, and that's based on the distance. Now we can determine DNA doesn't have north, south, east, west cardinal directions. The way we determine where genes are on the chromosome is by the crossing over frequency. All right. So when we look at where a chromosome is here, so let's say chromosome A is here, and it has it's likely to cross over 63% of the time in gamete formation to the other chromosome. We know that it's a certain distance away from the DNA, so we call this crossing over frequency map units. So this tells us that chromosome A would be 63 map units away from the centromere, and things closer to the centromere would have lower crossing over frequencies, so they would be closer, right? So something that, chromosome B is here, and it crosses over 27% of the time. It would be closer to the centromere, so it would be closer to the center of the DNA, and it would be 21 map units away. Okay, so that's chromosome mapping. So this is the last concept of Mendelian genetics, all right? So it's an extension on our independent assortment and segregation theories, which give us our Punnett squares, okay? So, how did we first figure this out? Well, this dude named Thomas Morgan was studying fruit flies. All right, so genes on the same chromosome are linked in some cases. Crossing over is a random event. And as you get closer, right, so the frequency of crossing over meiosis can be used to determine the position of genes, which is what I just said. So crossing over is a random event. But as you get closer to the centromere, because you would compromise the integrity of the chromosome if you broke up the whole thing to swap it. Alleles closer to the centromere are less likely to be swapped. Okay, so the further away, so they are from the centromere, the more likely alleles are to be swapped. So Thomas Morgan studied this first. So he studied fruit flies applying the concept of crossing over, and he came up with chromosome mapping. So here's what happens. In some cases, we expect certain ratios that come out of Mendelian genetics questions. But when ratios that are not predicted come out of Mendelian genetics questions, when we look at the offspring, we know that something weird is going on. So we know that segregation and independent assortment didn't occur. Okay. So when we get an unexpected ratio of offspring, we know that there's been crossing over, and we get what we call recombinant types. So we get recombinant individuals. Okay. So the first thing you need to know is the recombination frequency equation. Okay. So again, this just kind of walks you through in a textbook example, parental types and ratios that we should expect to find. So the recombination frequency, this is the simple way to look at crossing over. So your recombination frequency equation is the number of recombinants over the total number of offspring and that's going to be times 100%. Okay, so if I do a quick word equation, here it says, it's just really simple. In a brood of, 650 fruit flies, there was an unexpected number of white-eyed non-wild type fruit flies. There were 26 of these individuals, of these unexpected individuals. What is the recombination chocolate's done. Okay, so what is the recombination frequency of the offspring? Okay, so simple word problem. Okay, so we have how many recombinants? We had 26 recombinants. There's 650 total individuals. All right, that was how big the brood was. 26 were gave us unexpected appearances times 100%. And so your recombination frequency, I'm just going to put that as RF. Recombination frequency is going to be 26 divided by 650 times 100%. So your recombination frequency is 4%. All right. So that's the first way you're going to see gene, this type of gene combination. The more common type, though, is going to be a gene map. I'm going to give you a bunch of examples of gene maps. Okay, so I'm just going to wash my board off again. But you are going to get your Nelson textbook. I'm going to do a bunch of examples from Nelson. Okay, so in your Nelson textbook, page 641. Okay, I'm going to do a few of these. So I'm going to look at question 3, and then I'm going to do a bunch of questions from your key, and then I'm going to wrap it up. Okay, so Nelson, page 641, question number 3 says, create a chromosome map for each set of 3 genes for the given information. So here's what we've got. The crossing over frequency between A and B. So this is number 3. Okay, go outside. Okay, so the crossing over frequency between A and, I have genes A, B, and C in this question. Crossing over frequency between A and B is 23%. So we know that they are 23 map units away. Okay. The crossing over frequency between B and C is 11 map units. So we're 11%. So B and C is 11 map units away. And the crossing over frequency between A and C is 12%. And it's asking, right, so that's 12 map units. So it's asking me to build a gene map on this. Okay, so we know that the crossing over frequency is greater when the alleles are further apart. So I know that A and B are at my ends of my chromosome map. And that's because they're the furthest apart, right? So this is 23 map units. So this is how we do chromosome maps, right? We take the numbers and then build a puzzle. B and C are 11 map units away. So C is in the middle, but kind of closer to B. So B and C, and this is 12, and this is 11. And that adds up to 23, right? So our total chromosome map is 23 map units. So A, C, B. Okay. B on this question says, the crossing over frequency between gene X and Z is 8.5%. So this is B. So X and Z is 8.5%. So they're 8 map units away. Y and Z is 2.25 map units. Okay, 2.25 map units. And Y and X is 6.25 map units. And it asks you to make a chromosome map again. Okay? So X and Z are the furthest apart. So they're on my ends. That's 8.5. Y and Z. So Y is quite close to Z, right? So X, Y, Z, and where Y is closer to Z. And 8.5 minus 2.25 will give you 6.25. So this is 2.25, and this distance is 6.25 between X and Y. And that is your chromosome map. Okay, let's look at your key. Just going to erase my board again. Okay. Okay, so this is a very good question, page 94 on in your key, question 9 and question 10. And there's one more question I'm going to do. So you look at this whole gene map, okay, and this gives you the distance and map units of these different things away. So as this says, during meiosis, which of the following pairs of genes given above has the greatest chance of being separated by crossing over? So if we look at, remember that the further away, the more likely they are to be swapped. So if we look at M and D, so M and D are right here. Okay, so they're 12 map units away from each other. NE and P. So I added this up, right? So NE is here. Okay, and then P. So this would be 17 plus 16 plus 5. So 33, 38 map units. M and LC. So M is here, and LC is way at the bottom. So these are at the far end, so you know that the answer is C. So they're the furthest away from each other, so they're the furthest away from the centromeres. So they are very likely to be crossed over. Okay. 10 says, so you have this map unit. So, all right, the percentage equals map units. So this is the crossing over frequency between genes O and S. That's what we want to find. So it's going to be 31%, but I'm going to show you why. So this is question number 10 on page 94 of your key. So we want to find O and S. All right. Now, when we look at our map units, M, so if you look at crossing over frequency, M and S are the furthest away from each other. So they're on the ends. So M and S. And that's 64 map units. So this is 64. Okay, so those are the ends. Now what's my second largest one? That's the next one I'm going to go to. D and S. Okay, so D is 52 map units away from S. So I go from S to D. So D will be closer to M. And this is 52 map units. Okay. So I know that the distance between M and D is 12 then, right? Because I just subtract the difference. All right, so I know S is on an end. What's my next one? M and O. So M, and then O is 33 map units away from that. So O, right, so O would be kind of here. And this is 33 map units. So we want to find the distance between O and S. So that's what this is. Right? So I can find that now. And right, so I have 64 map units, and then I've got O accounting for 33 of that because the distance between M and O. So the difference is the distance between S and M and O, because O is in between S and M. So this is going to be 64 minus 33. And that's 31 map units. Okay? So the distance between M and O is 31 map units away. So they give you more information than you needed, right? So C is 31%. Okay? Is there one more question here? This one's a good one as well. I'm going to go through this one. So page 112. Question number 7. So the question says, the use of marker genes and the analysis of crossover frequencies of genes have enabled geneticists to map the location of many genes on human chromosomes. Blue color vision and blue color blindness are controlled by a gene on chromosome 7, the gene for the production of trypsin, and the gene responsible for cystic fibrosis are also found on chromosome 7. So the question is asking, so here's my chromosome map. The question is asking, which of the following gene maps shows the correct sequence of these genes? So let's make this. So I have, and then you have A, B, C, or D. Okay. So the marker gene is, so the marker and, this is question number 7. Okay, so the marker gene and the CF allele are 18 map units apart. Okay, so that's my furthest distance, so that's what I use first. Okay, just blowing through chalk today. All right, so that's the next one is the marker gene in tritanopia. Okay, so marker and tritanopia are 13 map units apart. So this is 13. Okay. So this is 18 minus 13 is 5, right? So this is 5 map units. So CF and tritanopia are 5 map units apart. Okay. Cystic fibrosis and trypsin are 6 map units apart. So I have to go from the CF up. So CF and trypsin are 6 map units apart. So trypsin is actually just above the tritanopia. And the two of these are 1 map unit, right? 6 minus 5 is 1. So it goes M, trypsin, tritanopia, CF, where the tritanopia is closer to the CF. All right? So the correct answer is A. Okay? So that is it for Mendel. There's one more question that I want to show you. And I've just got to pull it up first. Just give me a second here. There's one more multiple allele question that I would like to show you. And I always have to find it in my notes because I always forget where it is. Ah, here it is. Okay. Okay. So this question, you can find this if you just do a Google search of diploma exemplars. It's in the 2008 exemplars. This is a pretty classic question though. So this is a question, this goes back to dihybrid crosses. But you guys saw this with the black corn question that I gave you. But this says the coat color of Labrador Retrievers is determined by two alleles. The black allele, big B, is dominant to the brown allele, little b. So I'm going to write that down. So big B is black and little b is brown. Second pair of alleles, E, affects the expression of coat color. So in the homozygous recessive condition, little e, prevents the expression of black or brown. And then you get a yellow lab. So you get black labs from big B's, chocolate labs from little b, and then yellow labs when there's recessive E. Okay, so, right, so E stands for color expression. All right, so here's what we've got. So if you've got big B and a big C, then you get a black lab. If there's little b, and sorry, that's E, not C. If we get this, then you get a brown lab. And then if you have, doesn't matter what the B alleles are, but if there's E in the homozygous recessive condition, then you have a yellow lab. Okay. So the question is asking, if two Labrador Retrievers with the genotype big B, big E, little e were to be crossed, what phenotypic ratio would be expected in their offspring? Okay. So I'm going to put this on the board as well. And then that'll be it for the day. Okay, so we've got big B, big E, little e. So two heterozygous labs are crossed together. Okay, so that's going to give me these alleles. And then we fill it in. Okay, so the question is asking, how many are black? How many will be chocolate labs? And how many will be yellow labs? All right, so that little e indicates absence of color. So we count them up. So let's do the yellow and yellow chalk. So there's little e here, little e here, and there's little e here, and little e here. So there should be, right, black to brown to yellow. There will be 4 yellow. Okay. And then let's count black. So anything that's big B, big C, or big B, big E, I should say. So there's 1, 2, 5, 6, 7, 8. There's 9 of them that are big B, big E. So there should be 9 black labs. Okay. And then there should be little b, big E. That's what we're looking for. Okay. You're going to have, right, so little b, big E. There's going to be 3 brown labs. All right. So that's sort of the final dihybrid Mendelian genetics question that you're going to look at. This question will come in very handy when you do your Mendelian genetics test, by the way. Okay. So that's it for this unit. I will wait a couple days until I post another lecture so you guys can get caught up. All we have to do is molecular genetics now, so we are almost done. Okay, have a good one. Bye now.